encapsulated chamber basically lies on the same energy landscape as free solution, but it eliminates the complication of aggregation. So you could think of aggregation as making the landscape even more complicated and more full of kinetic traps. The idea behind the chaperonin chamber, in our view, is that it simply prevents aggregation from occurring and allows the Anfinsen cage essentially to function, to let the polypeptide find its way down the landscape without aggregating. Okay, so at this point in history, there were as many models for how GrowEL works as there were investigators in the field. And it seemed like the only way forward would be to get some sort of structural information about this molecule and be able to carry out functional and structure functional types of analysis. And I thank my lucky stars that Yale recruited this guy, Paul Sigler, from University of Chicago, and that I got sent over across town to talk to him about some trivial administrative nonsense that I can't remember anything about. But we talked for 30 seconds and we said, okay, forget this nonsense. And Paul immediately focused on chaperonins and said, how could these machines work? What's going on? Think of the beautiful symmetries. 2 rings, they're probably back to back. 7-fold radial symmetry. We've got to make crystals of this kind of molecule. And we said, absolutely. And that was the start of what was a 10-year wonderful collaboration that ended only with his untimely passing in the year 2000. So Paul dragged this guy in from the next room. This is now 1990, I think. Dragged in Zbyszek Otwanowski, who had been crucial to solving a number of structures in their lab. Actually started out as an astrophysics graduate student and was doing dishwashing in Paul's lab and kept looking over people's shoulder and solving structures. And so eventually he was recruited to the biophysics program at University of Chicago. So Zbyszek came in and looked at the basic story about the chaperonins and he said, well, the computing just can't handle this kind of problem at this point in history, but it's not a problem because it's going to take you guys 3 years to generate decent crystals of this molecule in the first place. And so that turned out to be correct. So Andrzej Jochimiak and I and a couple of my students went full blast to try to generate decent crystals of GrowEL. So we tried thermophilic versions because we thought they might be more stable. There were bad stories about E. coli GrowEL as a very badly misbehaved molecule that wouldn't crystallize. And we tried ultimately monomer versions of the E. coli molecule. We tried truncating it at either end and it still had ATPase activity. So we thought it was nominally folded in a reasonable way and we thought maybe we could crystallize monomers. All of that failed. But meanwhile, we'd become pretty sophisticated at overexpressing GrowEL. So compared to the original Georgopoulos strains that made GrowEL to the tune of about 10% or 20% of total protein, we now had these trick-driven plasmids that would make 95 to 98% of total E. coli protein as GrowEL. So you'd crack the cell and see just a single band on a gel. And so with that kind of ability, we could just make mutants ad lib basically and test all of them, purify them in 1 or 2 quick steps, set them up in trays. And so the winner of the competition was basically Kirsten Bragg, who was one of my students. She set up a whole wall of trays of a variety of different molecules. And the molecule that set up these nice orthorhombic crystals was amazingly a molecule that we just called wild type. It was just the second wild type molecule that had been produced by a PCR reaction, but it turned out that that PCR reaction had made a couple errors, and they were crucial. We don't understand even to this day exactly why they're crucial in producing nice packing, but it made this beautiful C2221 crystal in ammonium sulfate that diffracted to 2.8 angstroms. And so very quickly we went to the synchrotron, collected native data. Paul literally taught my lab how to do soaks with heavy atoms. And a single ethyl mercury soak really did the job with full occupancy of the 3 cysteines in each GrowEL subunit, which you could think of as being a real big pain in the butt to try to solve a structure with. But Zbyszek had already worked out a Patterson averaging program, as it were, that he put to work and he solved the structure in a single day. He then traced the chain correctly, amazingly enough, in 2 weeks. And that is amazing because these apical domains at the terminal ends of GRO-EL are somewhat disordered, have very high B factors, and he got the chain trace exactly right. It took us a year of refinement with Axel Brunger, Rashmi Hegde, and others to confirm that, but ultimately he really got it absolutely right from the start. So the molecule is 145 angstroms in height and roughly 135 angstroms in diameter. It's held together by these equatorial domains, both that make tight contacts both within a ring and across the equatorial plane. So that's the sort of base of the machine. And then connected to the equatorial domains, which have ATP binding sites on the inside, are these hinge structures that connect to the terminal apical domains. They're sort of flying in the breeze. They make just a single electrostatic contact with each other. And basically you can see here that they're not very tightly contacted. But they're the business end of the machine, because on their inside aspect is a polypeptide binding surface that I'll show you in the next slide. But what I want to say, though, is that there are really two central cavities of the machine, one upstairs here and one downstairs, because the C termini of the equatorial domains extend 25 amino acids that are not visible crystallographically, but can be seen by EM as masses of the roughly 10 kD in total mass inside the equatorial region of each ring. So basically you have two cavities that are separated by this massive stuff at the equatorial level. So we began to think about where the polypeptide binding site was. And I don't understand why, but initially we made mutations on the top of the equatorial domain, back here on the intermediate domain, and none of those affected polypeptide binding. Now, I should say that we can massively overproduce any mutant and quickly test it in vitro for whether it could bind a polypeptide. And then we looked at one of Helen Sable's early negative stain EM images, and we noticed that there was a blob in the apical hole of the machine. And it dawned on us that we're completely missing everything. The polypeptide binding surface must be the structures that line the cavity wall. And so we came in one morning after mutating all 9 of these residues to hydrophilic character and then putting those constructs into a GRO-EL deficient E. coli, and all of those E. coli were dead. So it became clear to us almost in one swoop that this must be the polypeptide binding surface. And indeed, we individually overproduced each of these individual mutants that were changed to hydrophilic character, and they were all dead for polypeptide binding in vitro. And so the point is, though, that for any one of these given residues, there's really 7 of them going around the ring. It's a continuous wall of hydrophobic surface that's exposed to solvent that's involved with binding non-native polypeptides. And that makes some sense because non-native polypeptides expose hydrophobic surface, and these contacts that would be water and entropy related types of contacts at a finer level would favor polypeptide binding of non-native states. But of course, as the protein folds to the native state, it buries its hydrophobic surface, and ergo, GRO-EL had no affinity for native states. And so we could ask the next level of question concerning polypeptide binding, which is how many apical domains do you really need to be functional in order to bind a non-native polypeptide, and what would that topology look like? So I was walking around on a beach on Martha's Vineyard with Tommy Kirchhausen talking about this problem, and Tommy suggests this completely loony experiment that we should just make a ring as a continuous coding sequence. Just attach all 7 subunits to each other, because then we could make any combination or permutation of mutations in the apical domains that would kill polypeptide binding by that individual apical domain, and we could then see what the arrangement is. What's the minimal arrangement required for polypeptide binding? So I convinced Christina, who I'll show you in a slide in just a second, to do this. It took her 4 or 5 months to make a 7-mer and then some of the mutants. But remarkably, when you express these molecules in E. coli, they made double rings. You could purify them, and you could characterize them. And indeed, what we found, to make a long story short, was that you need 3 consecutive good polypeptide binding domains in order to bind proteins like malate dehydrogenase or rubisco, which are sort of standard substrates that we use in this field. If you took 3 good subunits and broke them apart such that you had 2 consecutive good subunits on one side and 1 good one on the opposite side, that did not work very well. So it's the idea of some continuous hydrophobic surface that's critical for productive polypeptide binding. And more recently, in cryo-EM studies of malate dehydrogenase bound to GRO-EL carried out with Helen Sable's group, that's exactly what we saw, was MDH associated with 3 or 4 consecutive apical domains when bound in a GRO-EL ring. Interestingly, the binding was at different levels of the apical domains, some molecules more deeply than others. But again, this is an ensemble average of a huge number of molecules. Of course, what we really would dream about, and the free electron laser session last night was just off-scale interesting in this respect, would be of seeing individual molecules in single GRO-EL molecules. I mean, this is beyond just a nanocrystal experiment. This would be a single particle, single molecule type of experiment. But to actually see where the chain of a bound polypeptide is on GRO-EL at that kind of level would be a dream come true. Okay, so when a non-native polypeptide is bound to GRO-EL for any length of time, it really, in respect to being pulled up the energy landscape, occupies virtually a random coil conformation. So these are experiments that we carried out with Kurt Buttrisch, where we isotope labeled DHFR and offered it to a single ring version of GRO-EL, which is perfectly binding competent. And what we saw basically, whether we uniformly labeled or selectively leucine labeled DHFR, was that the polypeptide had no ordered secondary or tertiary structure. But it was productive because if you added GRO-ES and ATP back to it, you got the native state of DHFR back over